So, right. Thank you so much to the organisers for inviting me. It's been really good to actually talk to people and see the presentations today, because it means that part of my job is going to be easier, because so many of you know about EBI. Don't know how many people actually know about EMBL or anything like that. So we're going to go through a little bit of history there quickly and concentrate on at least 2 case studies around where we have promoted data sharing and open data access, and some of the future potential challenges that exist within biology as well, and where there are potential advantages and also areas where AI can play a huge role. So we'll go through those. No guess on the middle little picture there of what we're going to concentrate a little bit on, which has been dominating our lives for so absolutely long. So EBI, we're a world-leading source of public biomolecular data. Vision is to benefit humankind through advancing those scientific discoveries through the impact through bioinformatics, and we're part of this larger organisation, the European Molecular Biology Laboratory, which is Europe's flagship life sciences laboratory. Now, EMBL, we are one of 6 outstations. Our main headquarters are based over in Heidelberg. We have other outstations in Hamburg, Rome, Grenoble. I will say the Barcelona outstation, for those who have been anywhere near it, is possibly one of the most beautiful settings possible. It's right on the beach. If you ever get a chance to go there at all, please do. It's a fantastic venue. And also EMBL-EBI. Our missions: we want to perform excellent research, we want to provide excellent scientific services, provide advanced training to researchers, to students and visitors at absolutely all levels, to innovate and translate that technology into a commercial setting, but also to integrate those life sciences. Now, EMBL is slightly different from most other organisations. We are an IGO, therefore we are actually formed of 28 member states. We like to follow a little bit of the model of Eurovision, that you don't actually have to be within European continent to be a member of EMBL, as noted by Australia being an associate member state there. But those of you will notice that we are coming up to being 50 years old next year as well. So we have been around for a very long time. Now, EMBL-EBI's data services just cover pretty much every single aspect of biomedical research that you could possibly think of, all the way from fundamental chemicals, molecules and molecular and drug discovery, through to genomes, genes, RNA, through to proteins, imaging cellular structures, variation, epidemics, disease data, and also literature and knowledge management. Actually, in terms of the areas that I'm most involved with is over here in the genes, genomes and RNA section, specifically as PI of the Ensemble project, which is one of the main distributors of genomic data to researchers. Now, I'm going to go through a little bit of really what the EMBL-EBI model is. So firstly, we rely on researchers, funded researchers, to make discoveries, to generate data, and to want to deposit that data. And they probably have to do so in order to conform to some form of publication rights or so. For an example, genomic data, assembled genomes should be deposited with one of our databases or one of our peer databases before it goes to publication. So that makes its way into EBI as part of our archival systems. Now, we actually have 2 types of resources available. One of these is deposition databases, which is where that primary data goes. And then we have value-added databases. All the time, though, that originally primary submitted data is still freely accessible, freely to download. It also retains the rights of the original submitter as well. So it's not that we relicense that data or anything like that, but as part of the submission process, the researcher agrees to allow that data to be accessed again freely and to be reused freely as well. We then go back to those archives. So on that first slide, on that previous slide I showed, which went all the way through the stripe of biomolecular sciences, there were these deposition databases such as the European Nucleotide Archive, there was value-added resources such as Ensemble, and we are pinging all the way back into these archives because they're basically close to our compute. We can dip into there and we can actually retrieve data out, we can combine that, and we can actually form it together to actually analyze and enrich that data. And that then goes out into our value-added resources and distributed. And effectively, they become a form of truth in their own right. An example of this being the ensemble gene sets. So this is formed of analysis of transcriptomic data against genomes, and we perform that compute, we disseminate it, we accession it, and therefore it in its own right becomes a data set used that researchers do not have to go back and recalculate that information. And then new data, new designs, new experiments are performed on that data, and so this becomes a virtuous cycle of resubmission over and over and over again. We'll come back onto the reasons why that's actually a good thing as well for AI. In terms of the life cycle, the majority of our resources are in this establishing phase. There are resources such as the Bioimaging Archive that was moved down from, I believe it's Dundee it moved down from, or at least it's in collaboration with Dundee. But then we also have resources such as the PFAM database, which was about tracking protein families and domains, and also Array Express, which was the originator of, if anyone has seen an article or requirements around the minimal information for and insert an experiment name in here. Miami is one of the big ones, so that was originally from Array Express, but that was originally where you deposited microarray data to. So both of these have basically disappeared, but they've been subsumed by other EBI services. So we can continue to provide those services, but we do so in a more efficient manner. In terms of how we're funded, those member states do give us core funding there, but then we actually have anchor funding as well that comes in from external major contributors, and also we have project funders which allow us to actually boost ourselves beyond just our normal remit and to actually make advances as well. And I should say as well that UKRI fundamentally funds our technical infrastructure as well. So in 2022, the main thing to see here is that the numbers are basically big. 390 petabytes of raw data storage is probably one that highlight numbers there, which does always make me worry, especially as we talk about sustainability and the cost and the carbon cost of keeping all that going as well. However, though, when we start looking at the usage, effectively, we are exceptionally used. This is not live at all, promise. I never do live demos anymore, not since the incident whatsoever. But there was a definite jump in 2020, like a real significant jump of that. And it shouldn't come to any surprise that that correlates very strongly with the pandemic. And what's the best way of typifying this? I'm not sure that researchers had a lot of time to get papers out, but there was an increased effort to do something about the impending disaster that was all around us. And so we did see an uptick in that data. Now, in terms of usage, now we did a survey that was back in 2021 where we started to ask some fundamental questions about how well our resources are used to try and get some idea of value of how much costs can we save and how much do we actually enable that innovation. So 69% of respondees of one of these surveys said that not having access to our resources would have a major severe impact on their work, and 58% of those people said that they couldn't have done that research if they could not have got that data from anywhere else, and they couldn't have recreated it. So effectively, we were the only place you could get that from. Now, also, there's another way of assessing impact, and one of the other things that recently happened, this was basically at the beginning of this month, is that we ended up winning one of the Develts AI Prize. It's the 5th year it's run, and this was actually because of the efforts behind AlphaFold and AlphaFold DB, which will be one of the examples I'll go through a little bit more. But really, one of the things I want everyone to come away from this talk is that I fundamentally believe that EMBL-EBI is a stupendous place to maximize the potential benefits of AI because we have large amounts of open resources of data, large amounts of structured data, metadata, and biology fits this domain so well because there are so many problems yet to solve as well. So what else do we do? Well, we have co-developments and partnerships around the world. We have data sharing collaborators between the US, Japan, and Europe. We have new brokers that we're trying to build towards as well. There are new open data infrastructure models appearing as well. So perhaps some of the main ones we'll go through. No, sorry, that's a preview. There we go. the new global drivers, which is biodiversity projects and global pathogens, but then also alignment with a number of major global bodies, including the Global Alliance for Genomics and Health, but then also about how we support LMICs as well, so we don't leave them behind. Now, we are moving as well towards a transparent form of data licensing as well. There is a long-standing history in the sciences, in the biological sciences, of data sharing. Back when I actually started in this domain, which is far too long ago, the Fort Lauderdale Agreement was one of the very first types of agreement that appeared back in 2003, and that was basically pushing for pre-publication of genomic data. So the moment it was available, release it. Have some ideas around the restrictions in publication around that, but go for it. And then the Toronto Agreement extended that further to other biological data sets. So we do know that a number of our resources are under what is called the EBI terms of use, and we are committed towards clear and open licensing and know that that is basically our own in-house license. So we will start to move towards a CC0 license where applicable and try and homogenize this license as much as we can do across all of our resources. And we know this is important because of our own experiences with trying to reuse data under a myriad of different licenses is sometimes problematic and sometimes prohibitive as well to actually advancing our resources. So there's two things I was going to concentrate on. The first, which I think is, originally the title for this slide was the biggest open data sharing challenge we've faced. I'm not sure it really, it's hard to tell, but I think it was. That's my own personal view. So back in January 2020, this little virus started to appear. My wife booked for Christmas a holiday to Cologne for me, and then she said when she came home, it's like, yeah, I got you a Christmas present. It was last minute, but also I'm a bit worried about this virus thing that's kicking off over in China or so, and we all know where that ended up. But back in January 2020, we got the first record, what I would call the reference SARS-CoV-2 genome submitted into INSDC. If anyone has ever been into one of these databases, this will look familiar, but if not, this is effectively what the record is, and that is the sequence down there. That is the RNA, well, it's the DNA sequence, but SARS-CoV-2 is a single-stranded RNA genome. So a bit of a misnomer there. By March 2020, there was an obvious imminent need for us to concentrate resources around supporting SARS-CoV-2 research. It was so obvious, and having a diverse set of portfolio of services that I mentioned about before meant that we felt that we were a good place to do this. The problem is that the biology of the virus was complicated. I could probably spend about 5 minutes going through this slide alone, so I won't do, but there are a myriad of different things that happened in the virus and weird things that it does that still absolutely amaze me. But the fundamental problem here is that a number of our resources were not ready for this at all. We just did not think that we would have to move to viruses so quickly. But within 2 months, this actually was ready. But then the second question came up is, fine, we all now have these disparate resources, siloed data. How do you actually go about and integrate those together? Which led us onto the COVID-19 data portal. This is basically now, I would say, a complete success with 11 national other data portals available, and it aggregates information around the virus completely through one single unified interface of which researchers can access and download freely available, which is absolutely amazing. But we weren't the only show in town, because there was another group called GISAID, which was the global initiative for helping researchers to actually share data. And it was actually based on an influenza data database, which expanded itself to coronaviruses because it knew that that was, again, it was another really important thing for them to do. GISAID is incredibly popular because it has a very restrictive license in the background. To be part of GISAID, you basically sign up to GISAID, and then your secondary analysis is distributed within the platform. So you cannot distribute from without that platform without actually being part of it. So integration of GISAID-derived data is impossible into a platform like Ensemble because the licensing just does not permit it. And I have the emails to tell me exactly that that was not the case at all. However, though, this was really popular with researchers based in LMICs because there have been a huge number of issues around researchers being disadvantaged through early data sharing. We'll go through an example of that, but also attribution as well. So high-income countries basically coming in, taking their data, reprocessing, packaging, getting the publications for it, and that original LMIC researcher said, wait a second, that was my data, what are you trying to do with it? So just say bring some degree of assurances to that process. Now, we did actually have an open letter for saying that, you know, this goes beyond, this is a pandemic, we need to do more about this. I am one of those signatories. But ultimately, the problems don't go away. And they don't go away because ultimately, what we need in this domain is open data, but we need it with assurances. There's an article in Nature from this year that goes through a number of the issues and how problematic this area is as well, with a quote from our Institute Director, Ewan Burney. It doesn't go, you don't have to go back too far, so just to November 2021, with South Africa releasing its information on its new variant, and what it got in response to that was a complete shutdown for travel to that country. So what we're doing here, thanks for telling us about that there was this new strain flying around. It just doesn't quite correlate at all. So what we need to do is we need to enable this open sharing without necessarily punishing countries and scientists for being transparent and attribute that discovery to those researchers. However, though, we are still pushing forward with our own platform as well. And that in preparing for future pandemics, we have this new portal called the Pathogens Portal. All the pathogens on there are being selected from the UK Health Biological Agents and the World Health Organization's priority pathogens list. And this covers things from such as malaria, Plasmodium falciparum, to Lassa mammaria virus, which is hemorrhagic fever. And what we feel here is that basically what hampered us at the beginning with SARS-CoV-2 was a lack of infrastructure around being able to actually to bring together and to disseminate that information as well. So that's what this portal attempts to do. Now, to switch gear a little bit, AlphaFold has been mentioned a huge number of times. I'm going to go a little bit on a different angle when it comes to the AlphaFold developments, because hopefully I don't need to tell anyone how absolutely amazing this is. But really what actually helped to enable AlphaFold, or specifically what was it about EBI that helped AlphaFold succeed? One of those was the open data in the protein data bank as well, so 200,000 protein structures available as a training data set. Genomic sequencing had massively ramped up. We had over 200 million sequences in our UniProt protein database, and we also had another resource called Magnify that had 2.5 billion proteins. I'll go into what Magnify is soon enough. And just to underline about how much our data volumes are increasing and why, just to double down on this, that the depositions have increased massively, making EBI a really important place for, a really good place to actually center AlphaFold development around. But then also there was this framework as well, the CASP project as well, which meant there was suddenly a benchmark so that you could actually say, well, how good was that protein structure prediction? Now, Magnify, I said that that was actually one of the important parts of this. Magnify is a resource for understanding microbial communities, metagenomics. This is when you effectively go out into a field and you take a bit of soil and you sequence it and you see what's actually there, or you take a swab of someone's armpits. You would not believe how many people actually care about armpits, especially people that want to get deodorants that are really nice and stop all that bacteria from doing it. So there's a huge amount of metagenomic sampling happening from our guts to our skin to our environment. It's everywhere, absolutely everywhere. But suddenly now that is a huge repertoire of proteins out in the wild that we would not have previously retrieved if we'd gone out and sequenced every single genome as we are doing now. So effectively what this gave is a huge kind of wealth of life of proteins in the background that AlphaFold could basically pick into and use as part of its analysis. Now, one of the things that I sometimes doesn't get stated enough, so AlphaFold brought 200 million protein structures effectively to life. There was 200,000 originally in PDB, And this just allows us to start to answer weird questions like, can we start to understand disease resistance in bees? We could never get this protein structure at all. It just wasn't working. And now there's a database at EBI that you can just go in, download the structure. Fantastic, absolutely fantastic. There are other potential applications. Alpha Missense was another project recently released, which attempts to categorize the potential deleterious pathogenic consequence of variation in human genes. There is also now work on 3D structure homology, where there are proteins that basically fold into the same conformational shape. You would never notice that if you were just dealing with sequence alone. So AlphaFold is an absolute game changer. And it's a game changer that has been really reflected in just the number of citations and different publications that are coming back to that discovery. Okay, so what about our future? 3 areas that we are expanding into. But what I'm really going to just concentrate on are the 2 areas here, but specifically it's going to be this phrase here, pangenomes. I could have concentrated on biodiversity, and a lot of the items I'm going to talk about will be the same, and biodiversity being the idea that we go out and we sequence everything. Whatever you can see, we'll sequence it. Tree, fungus, insert anything else. There's a project called the Darwin Tree of Life that's coordinated out of the Sanger Institute. We're also partners on that project. There is another one called the European Reference Genome Atlas, the Vertebrate Genomes Project. Darwin Tree of Life is going to find 80,000 species. Vertebrate Genome Project is going to find 65,000 species. But fundamentally, what makes that possible is the fact that genomes are becoming cheaper to generate and easier to generate as well. And this gave me the opportunity to shove in this graph in, which I'm sure anyone that's been in one of these talks before has seen time and time again. Don't worry about it. It's just effectively, it's cheap, and also it's easy, and we've already seen pictures today of Oxford Nanopore's MinION, which is the size of a USB stick and can go anywhere. But I said I want to concentrate on pangenomes, which is really the next stage of genomics. Now, if we go back to the history of the Human Genome Project, the human genome is made of 12 volunteers. 11 of those actually came from Buffalo, New York. So 93% of the genome comes from 11 volunteers, and this is the advert that was posted for that, just there. And 70% of the genome actually comes from one individual as well. This massively fails to capture the diversity of us. We are not, well, I don't come from Buffalo, New York. I come from near Burnley for my sins. But there is this idea that we just didn't capture enough diversity, drove massive amounts of resequencing projects. And we know that fundamentally that there are alleles, there are differences that do have a disease impact. Cystic fibrosis, there are alleles that are specific to African subpopulations, meaning that you can result in misdiagnosis of cystic fibrosis because you're just not paying attention because just the genome wasn't exactly right. So I said pangenome's the next stage of genomics. Now, this is actually applying everywhere else as well. I mentioned about humans, but we can see that there are basically huge amounts of mice available as well. So these are laboratory strains of mice that are being used to create knockout models that are used in research. If we think about agricultural benefits of being able to track through particular traits from different pigs, again, quite important, and also just through all different types of farmed animals as well. But the Human Pangenome Reference Consortium, that is the next place that we're going to. And this is an effort to basically build the next stage. And effectively what you get down to is you have your current human reference genome, you have a number of different human genome references, and eventually you can put them into a graph-like structure, and it looks like a tube map, but effectively you have a way of representing all diversity within one given data structure, which is exactly where we need to be going on this. Now, one of the challenges faced in the moving into the pangenome, and the talk earlier was really good about, hey, you got electricity, what else can you do with it? Well, we actually do have these pangenomes, but the problem is that not many things actually know how to operate in this space. And the space is actually really quite complex as well because of the sheer quantity of different genomes in there. So We basically have workflows that only want to operate in linear space. And by linear space, I mean in these two spaces here, so the current reference genome and this genome. They don't want to work in this space because this space is hard. We need to ensure that sufficient diversity is actually captured within that genome. So again, no point in just going back to the same populations as we did do before. So this requires actually partnering up with multiple different pangenome projects. So projects that are in Singapore, which is an amazing project because that can combine first generation Chinese, Indian and Malay populations as well within one pretty nice open country. European Beyond 1 Million Genomes, which is a European project for resequencing. All of Us, which is based in the US, which is a resequencing project as well. So there are a number of different partners out there as well that we can engage with that will give different levels of open research and open access to that. But then actually, I think this is the most important point here, is annotating the novel genomic elements that pertain to each of these different subpopulations. Because again, if you think about the reference genome and that graph, again, there are components here that will be missing from the reference genome. And so we need to annotate those novel regions. Okay, so with that, thank you very much for your attention. Again, I'll repeat, AI is definitely a growing field within the biological sciences. We have excellent knowledge repositories and open access to data beyond just what EBI and EMBL do, but we do think our policies and our structured data and our metadata and our suite of resources just makes us this amazing resource of computable data. And if I think about challenges like the pangenome, I think about challenges around biodiversity, the numbers I talked about. I can't see how AI does not play a role in that future. And if I've whetted your appetite, we actually do have a conference in Heidelberg of next year as well. So please feel free to sign up, and thank you very much.